Dr. Karthik, who's an assistant professor in the Department of Bacteriology at the University of Wisconsin-Madison. His research focuses on microbial and viral ecology. He grew up in Mumbai, India, and earned his bachelor's degree in civil engineering at the National Institute of Technology. He did his doctoral research at the University of Michigan, studying the microbiology of hydrothermal vents with Dr. Greg Dick. He did postdoctoral research with Dr. Jill Banfield at the University of California, Berkeley, studying the microbial biogeochemistry of the terrestrial subsurface using high-resolution metagenomics. He's the recipient of several awards, including an NSF Career, the NIH Outstanding Investigator Award, and an ASM Early Career Award. Equally important, Karthik escaped the forests of France. Now, for those of you who follow us on Twitter, you know that one of our esteemed speakers got lost while hiking in France. That would be Karthik. Without a mobile phone, his well-being rests in the hands of his friend, who it turns out provided such a poor description of Karthik to law enforcement that there's no way they ever would have found him. So thankfully, he made it out on his own and joins us here today, straight out of the forests of France? Is that right? Okay, to share his research. So Dr. Karthik, welcome. The floor is yours.
All right, thanks for that wonderful introduction, Pete, and thank you to all the organizers for inviting me. It's a pleasure to be here and have the opportunity to talk to you a little bit about some of the work that I do. So I'm an assistant professor in the Department of Bacteriology at University of Wisconsin-Madison, and my lab's interested in a lot of things, but for today, I'll talk to you about viruses in microbiomes. As just right at the beginning of the talk, I'll clarify two things. One is that throughout the talk, you'll see that I use the words viruses and phage quite interchangeably. Although I'm using them interchangeably, for the most part, my lab studies phage. We are interested in all viruses, but we are very partial to phage. And when I say microbiomes, of course, we're interested in microbiomes, but the kind of data that my lab deals with is primarily metagenomic and metatranscriptomic data. We're not really dealing with other forms of omics data. So as far as the, okay, what's going on? Okay. So just to give you an outline of this particular talk, I'll just give you a brief background on our laboratory, our very diverse interests. I'll give you a little bit of a primer on viruses, phage, how we can go about studying them, why they're important. And then I'll spend most of my talk talking to you about virus bioinformatics, so some of the bioinformatic approaches and software tools that my lab has developed to enable the study of viruses. And then towards the end, I'll show you how we can track viruses and their impacts on metabolism. And metabolism is a particular interest of our particular laboratory. So my laboratory really works at the interface of three different fields. We are very interested in biogeochemistry, microbial and viral systems, ecology, and computational biology. And the fundamental question that really motivates us is trying to understand the roles of microbes and viruses in driving sulfur metabolism. So we're very, very partial to the element sulfur. You'll see that. And in turn, how sulfur metabolism and metabolism in general impacts diseases as well as ecosystems. We have very diverse interests spanning both environmental as well as human microbiomes. We work all the way from the deepest depths of the oceans in hydrothermal vents, where we do our own sampling. So we go out on cruises. We are a field-going lab, all the way to freshwater lakes. I mean, at the end of the day, my lab is in Wisconsin. We are surrounded by lakes on all sides, so we've sort of expanded and diversified into also studying freshwater lakes. And slowly, my lab's also diversified into studying the human microbiome. And specifically, what we are interested in now are certain diseases and disorders that are associated with sulfur metabolism, the primary one among them being colorectal cancer. What's interesting about sulfur is that depending on where you are in the environment or what ecosystem you're working in, different forms of sulfur and different kinds of sulfur transformations dominate the ecosystem. So for example, in hydrothermal vents, the kinds of transformations you will see are primarily inorganic sulfur transformations, primarily sulfur oxidation that dominates chemolithoautotrophy in hydrothermal vents. If you look at, say, freshwater lakes, you'll see both organic and inorganic sulfur transformations. And if you're thinking about the human gut, organic sulfur transformations dominate this particular ecosystem. Our laboratory also tries to enable the analysis of microbiomes. So we are also developing software tools. These are some of the tools that my lab has put out. The majority of these are associated with enabling the study of phages. So these are 3 tools, Vibrant, Propagate, and VRAM, that I'll talk to you about today. But we've also developed Metabolic, and this is a software tool that can enable metabolic as well as biogeochemical analyses in microbes. And much of these tools are centered around using genomic or transcriptomic data. Now, thinking about viruses, why should we care about viruses or phage? Viruses and phage really are all around us. They are the most abundant entities. And if you're thinking about their applications, they're really important in the context of human health, biogeochemistry, ocean health, all the way down to the food industry. They have a wide variety of uses and applications. But from the perspective of our interest, my laboratory's interest, we are most interested in viruses and their association with human health and biogeochemistry. If you're thinking about the life cycle of viruses, you can take a rather simplistic approach, and you can say, okay, what do viruses do? Viruses land on an organism or a cell. Let's imagine this is a phage. It's landing on a bacterial cell. It infects the bacterial cell, it replicates, eventually the bacterial cell dies, the virus propagates. So it's a rather simplistic view, but it's a relatively accurate and fair view. But why is this important? If you're thinking about it, this is a very, very abundant process. And just to put that into perspective, if you looked up at the night sky, there's about 10 to the power 22 stars in the entire universe. The night is full of stars. And just in the oceans, every second, there's 10 to the power 23 viral infections. So that gives you a sense of the magnitude of viral infections that are happening, the timescales that are involved, and how important viruses can be by their sheer abundance. So when you're thinking about viral processes, abundance of these processes is a very important metric to take into account. If you're thinking about viruses in ecosystems and the kinds of roles that they play, viruses really are the most abundant biological entities in every environment. And this is somewhat of a cliched argument now, but it's really important. If you're thinking about the oceans, and if you were to quantify all of the biomass that's present in the oceans, viruses would only constitute about 5% of the biomass. But now if you turned it around and said, let's count the number of organisms and let's look and see the abundance, 95% of all biological entities in the oceans are actually viruses. So it gives you a sense as to how abundant they are. And by the sheer abundance and the processes that they mediate, they can control the availability of nutrients, they can influence microbial community composition, they can kill the most abundant organisms, they can turn over microbial populations, and they can also drive metabolic processes. And this is something I'll talk to you a little bit about. Viruses typically come in a variety of shapes and sizes. This is probably some of the most common shapes that you would have associated with. And these are typically double-stranded DNA viruses that infect bacteria that look like this. But typically, viruses do have a wide variety of shapes. Now, how exactly do viruses impact biogeochemistry and nutrient cycling? So there's a few different processes. But when a virus comes in and infects a cell, there's 3 steps, or say 3 particular ways in which it can impact biogeochemistry and nutrient cycling, both at the scale of individual cells as well as the ecosystem level. The first one involves what are known as auxiliary metabolic genes, or AMGs. And I'll tell you a little bit about what AMGs are and why they're important. But AMGs essentially are used to manipulate hosts. So that's AMGs in a nutshell. But viruses can also manipulate hosts in other ways. They can mess with transcription. There's a number of ways in which they can impact and manipulate their hosts. And finally, when a virus kills a bacterial cell or an entire population, what it does is it releases intracellular contents. And these intracellular contents can be elements, it can be compounds. So this can then go in and impact the microbial community, because all of these now are available to the rest of the microbial community to utilize and turn over. Now, thinking of this from an ecosystem perspective, or more specifically a human gut perspective, why should we be studying viruses in the human gut? For the most part, when you think about human microbiome research, we have sort of forgotten about the viruses. Most human microbiome research really has focused on the role of bacteria specifically in human health and disorders. But now there's increasing recognition that viruses are important components of the microbiome. And at the end of the day, viruses are equal, or if not, very important members of the microbiome that are interacting with the host genome, that are eliciting host immune responses, and that can then go on and either potentiate or protect against diseases. But the key takeaway here is that in the grand scheme of things, we know little to nothing about the role of bacteriophages in the etiology of most diseases. All we know is that they're there. We don't know why they're there, and we don't know what they're contributing. And that's something we hope to contribute towards. Now, I mentioned that viruses can manipulate microbes through what's known as viral auxiliary metabolism. So what exactly is viral auxiliary metabolism, and why is it important? So auxiliary metabolic genes, or AMGs, as I'll refer to them through the rest of the talk, are acquired from their hosts. They are retained on the viral genome. They are functional, and they offer fitness benefits to the viruses. The phages typically use AMGs to exploit host metabolic niches. And I'll give you some examples through the rest of the talk. These AMGs are not involved in essential phage processes, such as replication or lysis. They typically are involved with metabolism or other examples, or say other processes. There's two very broad groups of AMGs. These are groups that we've sort of come up with ourselves. So these are not formally defined, but if you look at it, there's two broad groups of AMGs. So if you think about it, group one would be AMGs that are associated with increasing host metabolic productivity for phage benefit. So these are typically AMGs that are associated, let's say, with creating energy. Okay, so this could be nitrogen, carbon, or sulfur metabolism. So if you look at, say, hydrothermal vents, you would see sulfur metabolism through the rest of the oceans, especially the sunlit oceans. You can see photosynthesis AMGs. And these AMGs are associated with production of energy, which is then sort of funneled into production of viral particles. So essentially, there's a direct increase in virion production, or viral particle production, that can come about from these AMGs. The second group of AMGs that I would argue are far less studied or very poorly understood are group 2 AMGs, which are associated with enhancing host survival for phage transmission. So these can be associated with virulence genes, sporulation, toxin, antitoxin systems, stress responses, just to name a few. Okay. But essentially, these AMGs alleviate stress in the bacterial cell when it's infected, and they help the cell to stay alive a little longer, and essentially lead to an indirect increase in virion production. Okay. So the first ones are associated with direct increases in virion production. The second one's associated with an indirect increase in virion production. So the big picture question, why do we study viruses? We are interested in understanding the roles of viruses and virus-driven metabolism, specifically using AMGs, in modulating microbiomes in the environment as well as in the context of human health. And my lab's primarily a lab of microbial ecologists, and we are interested in studying viruses from three distinct perspectives. So we want to study viruses through the lenses of diversity, distribution, and ecology. So when we're thinking about diversity, the kinds of questions we ask are, how can we accurately identify and characterize viruses from microbiome data? When we're thinking about distribution, we want to try and see if we can use viruses as predictive markers of, let's say, human disease, or of ecosystem health, or even, let's say, biogeochemical processes. And finally, from an ecological lens, we want to try and understand the roles of viruses in driving gut dysbiosis or biogeochemistry in nature. So with this sort of background, let me go in and start talking to you about some of the bioinformatic approaches that we've developed to study viruses from microbiome data. And just to remind you, when I say microbiome data, we're primarily dealing with some kind of genomic or transcriptomic data, typically metagenomic, genomic, or metatranscriptomic data. So very early on, what we started to do was try and understand genomic signatures that are present in the genomes of different kinds of organisms. And what do I mean by genomic signatures? So these can be something as simple as GC content to, let's say, gene-specific features, like how many genes are present in which strand of the genome, to gene coding density. So certain features like this. And when you begin to synthesize all of these features together, what we realized is that you can use these signatures to tell different organisms apart. So what you're looking at over here is a plot, a PCA plot, of gene signatures, or say genomic signatures associated with different kinds of organisms. So the viruses are in green, plasmids are in red, and bacteria and archaea are in blue. And you can even put eukaryotes on here. You can put other kinds of genomes on here as well. But the gist of this is that when you use genomic signatures, you can differentiate the viruses from other kinds of genomes. So for example, the viruses cluster very distinctly from both the plasmids as well as bacterial and archaeal genomes. And this underlying framework to differentiating viruses, we actually built it into a particular software known as Vibrant. Vibrant is a software that can use metagenomes or metatranscriptomes and identify viruses by what we call iterative annotations. Essentially, it's using protein annotations and the virus-like nature of protein annotations in a genome to identify different kinds of viruses. If you're thinking about what Vibrant can do and what is the purpose of Vibrant, it can identify bacterial and archaeal viruses from metagenomic and metatranscriptomic assemblies as well as individual genomes. It's built for all kinds of viruses. So double-stranded DNA viruses are the most abundant kind that we typically encounter in different kinds of samples, including in the human gut as well as the environment. But we can also work with single-stranded DNA viruses or RNA viruses. We can also work with viromes, which are essentially samples that have, where viruses have been concentrated, or we can work with mixed or bulk metagenomes. Thinking about what Vibrant actually does and what can it help you with if you have microbiome data, to begin with, it can identify lytic phages from metagenomes. So these are free-living viruses that don't have an integrated cycle. The second, it can identify prophages. So these are viruses that have integrated into microbial genomes. So it can also identify these viruses in microbial genomes and excise these viruses. Third, it can annotate phage genomes and help you understand how good your phage genomes are. So to give you a sense, it'll give you a sense of phage genome quality. For example, do you have just a partial genome fragment? Do you have a complete genome? Do you have a circular genome? So this is something that you can get information about. It can also identify auxiliary metabolic genes in phage genomes. And finally, it can help you visualize some of your data. What is really unique about Vibrant and what we are particularly excited about is the fact that it can identify auxiliary metabolic genes very accurately. It can do this both at the resolution of a single viral genome as well as the entire ecosystem. So if you're interested, let's say, in one virus genome, let's say you're interested in viruses that infect cyanobacteria in the oceans. Here's an example of a cyanobacterium in the oceans, and let's say there's a virus that has genes associated with photosynthesis, that it's manipulating the cyanobacteria. You can actually identify the photosynthesis genes and then use these to undertake further analysis. But what you can also do is get a sense of the entire viral community and all of the auxiliary metabolic genes that are present in the entire viral community. So this gives you a sense of how viruses and viral communities as a whole are manipulating microbes in an ecosystem. So for example, here is an example from the human gut. So you're looking at a human gut virome. And all of the auxiliary metabolic genes have been identified by Vibrant, and Vibrant's sort of characterize these into broad metabolic categories. So that's what you're seeing over here, the broad metabolic categories over here, and the number of AMGs that have been identified on the y-axis. So what you can see over here is that the largest fraction of AMGs in this human gut virome are associated with amino acid metabolism, followed by, let's say, energy metabolism and cofactors. Now, if you were to look at a different system, so for example, soil or hydrothermal vent ecosystems, I've studied these, so I can tell you that, for example, in soil, you would see that the greatest number of AMGs would be associated with carbohydrate or sugar metabolism. If you went to hydrothermal vents, the greatest number of AMGs would typically be associated with energy metabolism. So every ecosystem will have a distinct signature associated with viruses and their AMGs. So thinking of some of the real-life applications about where Vibrant's been used, and I'll just highlight some of these. But so we've used Vibrant in hydrothermal vent systems to identify viruses that infect sulfur-oxidizing bacteria. It's been used to identify viruses that infect archaea, specifically Asgard archaea or Loki archaea, the archaea that are closest to eukaryotes. It's been used in soil. And most interestingly, through a collaboration, we were able to study viruses that infect patients, or say cohorts associated with rheumatoid arthritis. And this is particularly interesting because even in rheumatoid arthritis, what we found is that if you're comparing cohorts of healthy patients with patients who have, let's say, rheumatoid arthritis, you can see that they harbor different viral communities, and they also harbor, and the viral communities have different AMGs, and that's essentially what you're seeing over here. Okay, that was Vibrant. Now let me move on and show you two other softwares that we have developed. And essentially, these softwares will now, so imagine that with Vibrant, you can actually identify viruses from microbiome data. But once you've identified viruses and characterized them to some level, what do you do after that? So now let me show you two different kinds of softwares that will help you characterize viruses from microbiome data better. So the first one is Propagate. Propagate works exclusively with prophages. Propagate stands for Prophage Activity Estimator. So essentially, it identifies prophage populations that are actively replicating or propagating. What does this mean? So imagine this scenario. When you have prophages and you wish to track whether these prophages are active or dormant, we need to understand these stages of infection. And with prophages, there are different stages of infection. So it starts with, let's say, prophages that are dormant. So a prophage is integrated, let's say, into a bacterial genome. So what you're looking at over here is the bacterial genome in pink and the prophage genome in green. So the prophage is integrated into the bacterial genome. Once the prophage begins to activate, you will see that there are a greater number of copies of the phage genome that are produced. So here you see the phage genome has been replicated. After this, once it enters a mid-lytic stage, the prophage begins to assemble particles, and eventually this is a full-blown viral infection. The cell dies, and the viral particles are propagated. How does this actually look like if you're looking at it from a genomic lens? If you're looking at sequencing data and looking at genomes, how does this actually look? So if you have an active prophage infection, so if you have an active prophage, there will be many greater copies of the prophage genome than the host bacterial genome. So the host bacterial genome, again, is in pink. The prophage genome is in green. So you can see that the ratio of prophages to host genome, in this particular case, is about, the ratio is about 6:1. Now, if you had a dormant prophage, that is, it wasn't active, the ratio here would be 1:1. So this information, okay, with regard to genome coverage is something you can actually observe. If you looked at genome coverage, if you had a dormant prophage, the genome coverage over the length of the genome is going to be relatively stable. On the other hand, if you knew exactly where your prophage was on a given bacterial genome, and the prophage was active, this is what you will see. There will be increased coverage associated with this prophage. And this information and the effect sizes associated with this can be utilized to identify whether a prophage is active or not. And let me show you an example to illustrate how Propagate can actually help you analyze your viral data, or prophage data specifically. What you're looking at here is a spatial series of soil samples and prophages associated with one lineage of bacteria. The bacteria are Myxococcales. So these are different. Each of these is a different soil sample, and all of these are the same Myxococcales host. But as you can see, what's happening with the prophages is that prophages indicated by these red dots is that in some samples they're very active, in other samples they're not active. And once you identify in which samples prophages are active, which samples they're not active, you can go in and look at either spatiotemporal characteristics or let's say biogeochemical measurements and try and understand the stressors or the chemical signatures behind why prophages are active in one particular sample versus another. And this is important both in the context of human health as well as environmental systems. Now, the third software I'm going to talk to you about is the latest one that we have developed in our lab, and this one's called VRIME. VRIME is something that can enable virus binning, and this is something we are very excited about because this is something that was not achievable and something that was not possible even So before I sort of get into that, what is virus binning? What is binning, and why am I excited about this? So if you think about it, binning refers to the process of reconstructing genomes. So let me explain what this means. Every genome, when you're reconstructing genomes from metagenomic data, is a puzzle. So imagine you have a puzzle, and I already tell you that, okay, this puzzle comes from one genome. So I've taken one genome, broken it into different parts, and now the objective is to put this back together. It's a puzzle. And the objective of this puzzle is to create what a MAG or a VMAG. What does that mean? It's a metagenome assembled genome, or in our case, a VMAG stands for a viral metagenome assembled genome. Now, if you bind this data accurately, this particular puzzle will come back in as one viral genome. So then, if you're thinking about the implications of this, this means that we have reconstructed one virus from this data, and it's an accurate representation of the genomes and an accurate representation of the diversity associated in your sample. However, if you did not bin your data, what you could end up with is something like this. In this particular case, there are 8 different fragments. Typically, you would assume that there are 8 different viruses in your sample. And this results in fragmented genomes and a distorted perception of diversity in your samples. So while you actually only have one virus, you assume that you have 8. And this is something that was common in viral metagenomics that we hope VRIME can address. And what does VRIME do? VRIME uses machine learning to enable genome binning. So we use a highly curated reference data set to learn signatures associated with how fragmented genomes can actually be put together and implement this into a supervised machine learning framework. So essentially, imagine this scenario. You have fragmented genomes that look like this. You curate these. So you remove some of these fragments. Let's say the green ones come from bacteria. So we can remove the green ones. And we know that the purple ones come from one virus. The orange ones come from another. Eventually, we end up with genomes that look like this. So we can put that together using supervised machine learning. And once implemented in a full-fledged metagenome, it can actually produce really good signatures. So for example, what you're looking at over here is just a network diagram that is connecting different parts of genomes. So for example, these dark blue dots that are connected come from one virus. So there are different fragments of one virus that we've managed to cluster together. So some of these represent large genomes. So for example, if you focus on this orange one or the blue one, these are large viral genomes. So possibly viruses that infect eukaryotes or double-stranded DNA viruses. But on the other end of the spectrum, we can also work with really small viruses. So single-stranded DNA viruses would be really small, something like this, or even RNA viruses. What is the framework behind VRIME? VRIME uses read coverage. So imagine the scenario. Let's say there's 2 viruses in a data set. There's viruses shown here in orange and viruses, let's say, shown in blue. Imagine that these 2 viruses are abundant to different levels. So one of them is more abundant, one of them is less abundant. So the orange one is more abundant, the blue one is less abundant. And you can use the information and the coverage effect sizes in here to help with binning virus genomes. And you can also use sequence features that are encoded in the genomes themselves, so such as formers, codon usage, GC content, etc. And all of this actually feeds in to a supervised machine learning model and a certain amount of iterative clustering that eventually leads to beautiful bins. So you start with something like this, but it's all mixed together, and then eventually you will end up with really good individual viral genomes. And to give you a sense of an example and why this is really important, here's an example from a skin microbiome, viruses that infect streptomyces. And here you have a really clean reference genome. So it's a genome of a virus that's been isolated. You know it's in one fragment. You know exactly what the genome looks like. And here is what you were able to recover from a metagenome. There were 4 different fragments. We would have, without virus binning, we would have imagined that there are 4 different viruses instead of 1. But because we can bin these, we now know that they all come from the same particular viral genome. But there is one other reason why this is really important. In the context of what AMGs mean, as an example, sometimes when you have fragments that look like this, and let's say there's 3 genes over here that look like metabolic genes, often you would tend to ignore them thinking this is a prophage genome. For example, these genes could have originated from the bacterial genome itself rather than being on a viral genome. But when you bin genomes, and you're able to put fragments together, you have a greater context for individual genes. And that's the real power of using something like this. So with that discussion of virus bioinformatics and some of the approaches that we've developed in our lab, the rest of the time I'll spend talking about viruses and metabolism. So I'll give you some examples of how we've utilized some of these approaches and studied the role of viruses in different environments, specifically with a focus on sulfur metabolism. So I've mentioned that we have a huge interest in sulfur metabolism, both from a microbial as well as viral perspective. And we are very interested in understanding the specific roles of viruses in driving sulfur transformations, both in the environment and in the human gut. And this is work that's driven by 2 PhD students in my lab. Chris Kieft, who is a 5th-year PhD student about to graduate, and Elise Carley, who's an MD PhD student in my laboratory. And by screening publicly available viral genomes, what we were able to determine is that there is an enormous diversity of sulfur genes, AMGs, that are encoded by viruses. And what are these AMGs, or what kinds of transformations do they encode? The entire spectrum of organic and inorganic sulfur transformations. So as an example, when it comes to reduction of sulfate all the way to sulfide, or say oxidation of sulfur compounds to sulfur, transformation of organosulfur compounds, amino acids, amino acid derivatives such as taurine. So the entire gamut of organosulfur compound transformations can be done by viruses. Not all of these are encoded on the same virus. Every virus typically will encode maybe 1 or 2 of these. But the reality is that there's an enormous diversity of sulfur AMGs that are encoded by viruses. When you begin to look at these viruses and you try and understand if there are any similarities, it turns out there's actually a lot of diversity. These viruses often don't have anything in common. So these viruses can infect bacteria, archaea, eukarya. If you look at what kinds of organisms they infect free-living organisms, host-associated organisms, pathogens. They are present in different environments, soil systems, marine systems, hydrothermal systems, the human gut. They have different genomic arrangements. Some of them are linear genomes, some of them are circular. Where they are present on the viral genome also varies a lot. And they belong to very different viral families, so an enormous amount of diversity overall. If you're thinking about genes specifically associated with organic sulfur degradation and production of hydrogen sulfide, we notice that these are particularly abundant in humans. No matter where you looked at in the human microbiome, whether it was the oral microbiome, the gut microbiome, or the vaginal microbiome, these genes are very abundant. But what's interesting is that different genes are abundant in different microbiomes. Now, one interesting theme that did come out of this is that a lot of the organosulfur transformations that viruses can sort of drive, some of them do lead to the production of hydrogen sulfide. And this had us very intrigued because hydrogen sulfide is a very important compound, both in the environment. It is the most reduced form of sulfur. So microbes can utilize this as for energy metabolism. But from a human perspective, it's really important in the context of human health. It's genotoxic, it's pro-inflammatory. So there's a lot of important reasons as to why hydrogen sulfide production is very significant. But what is the importance of hydrogen sulfide in the context of viruses? Why would viruses drive the production of hydrogen sulfide? So we ran a very simple experiment to test the impact of hydrogen sulfide on phage. And the experiment involved increasing hydrogen sulfide concentrations and looking at the number of phage particles that were produced. And this involves a model lactococcus lactus and lactococcus phage system. And the message here really is that more viruses are produced under higher H2 concentrations. As you increase the H2S concentrations, the number of viral particles that are produced from an infection seems to increase. Why is that? We are not able to explain right now. We don't have a mechanistic reason for this. But it's clear that there's a trend here. There's a statistically significant trend associated with increasing H2S concentrations and a greater number of viral particles. If you're wondering, what happens to the microbial hosts, for the most part, the microbial hosts are completely unaffected. Right? It doesn't impact them whatsoever. So on the basis of this, we have a model for what we call virus-enhanced sulfide production. I'm going to focus on one specific process, which is cysteine degradation. So cysteine desulfurization can produce H2S. So there are genes for cysteine degradation in viruses. And when these viruses infect bacteria, it can cause uninhibited cysteine degradation in infected host cells, which then leads to enhanced viral replication. So what does this look like? Let's say there's a bacterium. It has very high intracellular concentrations of cysteine. Typically, cysteine concentrations are on the order of millimolar. When a phage comes and infects this bacterium, imagine that this phage has genes for degradation of cysteine. One particular set of genes are cis-K. So the virus would go in, express cis-K inside these host cells, and drive the degradation of cysteine. Bacteria, the infected cells specifically, are forced to upregulate their cysteine metabolism to produce H2S, which causes viruses to replicate inside the host cell. And the H2S, this is interesting. The H2S can then go on and cause intestinal inflammation. So if you're thinking of it from a human health perspective, we've been very interested in colorectal cancer, and we've been studying the association of sulfur metabolism with colorectal cancer. And what does H2S do in this particular environment? H2S is associated, let's say, with organic sulfur metabolism coming from cysteine and methionine. And it's a key mechanism that links Western diet that's rich in, say, proteins and meat, and that's where a lot of the cysteine comes from, and CRC risk. If you think about H2S, in a healthy gut, H2S can come from, let's say, sulfate-reducing bacteria, taurine-respiring bacteria, or cysteine-metabolizing bacteria. But the concentrations of H2S are micromolar, so very, very low H2S concentrations. But if you had colorectal cancer or a different gut disorder, then the observed concentrations of H2S are actually millimolar. Where is this H2S coming from? A large part of this is actually coming from bacteria that are degrading, let's say, either cysteine or methionine to hydrogen sulfide, or say other compounds to hydrogen sulfide. But what if phages also played a role in this? And this is something that we haven't tested yet, but we think this is a possibility. So if you had a healthy gut, all of the bacteria, if you had a healthy gut, there's not much H2S here, there's no inflammation. But if you had bacterial H2S in production, there can be inflammation. And eventually, if there's both bacterial as well as phage H2S production, then you can have greater amount of, say, inflammation that can impact, say, progression of certain diseases, specifically colon cancer. Okay, in the last few minutes that I have, what I thought is I'll show you some really interesting new data that we have and just talk to you about an approach that you can use to study and track viruses and phage in the environment. This is something that is relatively new, but we are very excited about. So we've gotten into these endeavors where we are tracking viruses using time series metagenomics in two different environments. We are using this in hydrothermal vents and the pelagic oceans. and in freshwater systems. And I'm not going to show you data about hydrothermal vents today, but I'll just briefly show you some data about freshwater lakes. We have a beautiful model system in Lake Mendota, which is on the bank. I mean, the University of Wisconsin is on the banks of Lake Mendota. And we have a 20-year time series of Lake Mendota. What we have is an integrated 12-meter sample that we've conducted, that we've collected from Lake Mendota every week for the last 20 years. And because it's a year-long sampling session, we have samples that span the entire 12 months, so all the way from January to December. And because Wisconsin is a temperate environment, there's seasonality. And this allows us to study seasonality associated with freshwater viruses and bacteria. And what's interesting is that we can study the diversity, distribution, and ecology of freshwater systems. And just to give you a snippet of this vast data set, we have in total over 465 metagenomes. So it's a huge amount of data. So far, we have over 1 million viruses. And what does this mean? What do 1 million viruses from this freshwater system mean? So if you're thinking of viral species, the largest public data set of viruses today has about 900,000 viral species. But from a single environment, just because we have such a long time series, and that spans different time points, we have about the same order of magnitude number of viral species. So we're about to increase the amount of viral diversity present in some public databases by almost double. We've produced metagenome assembled genomes from these systems. And just to show you a snippet of this data, in the context of diversity, we have found that the most abundant viruses and AMGs are distributed across almost all seasons. So genes that are associated, let's say, with photosynthesis or some of the most abundant processes, you will see a seasonality, but they're present, more abundant in the summer, but typically they're always present. If you're interested in very specific organisms, such as, let's say, cyanobacteria, these nostoc cyanobacteria or cyanobaceae, you'll see that different viruses infecting a single host have different distributions. But what's also interesting is that here we have strain-level variation associated with the viruses. So in a single viral species, you can have some viruses, some viral strains that have AMGs, and some that don't have AMGs. And there's different kinds of seasonality you will observe with these. So for example, viruses that have AMGs may peak in the summer. Viral strains in a particular species that have AMGs may peak in the summer, and some others may peak later in the fall or the winter. So with that, I'll just summarize what I've spoken to you about today. I showed you three different tools, so Vibrant, Propagate, and VRIME, that can enable characterization of viral ecology. I showed you data about viral metabolic genes that are associated with a variety of sulfur-associated processes. If there's one takeaway from viruses, you can imagine that hydrogen sulfide can increase viral fitness. And finally, time series viromics is a powerful approach to study viral diversity, ecology, and evolution. I'd like to thank my funders. Much of this is funded by the NIH and NSF, but also USDA, JGI, the Schmidt Ocean Institute that has funded our cruise, and WRF. And I have a whole host of collaborators in the human microbiome, freshwater, and deep sea research, without whom much of this would not be possible. And I thank all of you for listening, and I'll take any questions.
Terrific, Karthik. We've got time for a question or two. Yes, back in the corner. Maybe you could stand up and just temporarily take off your mask to project.
So if you have time, I have two related questions, which has to do with the vibrant annotation pipeline.
Okay.
So with respect to the way you can BCA separate out genome based on various markers, for either AMGs that are recently acquired, or in the case of temperate phage, do you actually see that they are more like the host or somewhere in the middle of host to lytic transition? Right? Because you should be able to track, maybe, I guess I don't know the resolution with which you can track for individual genes compared to full genome.
So I don't think you need to use Vibrant to do that. But if you did want to track AMGs and find out whether they were recently acquired or, You could do it. I mean, it's not straightforward what you're proposing, but you could do it. As far as AMGs are concerned, there's a spectrum associated with their similarity with hosts. They can go all the way from 50% protein identity to 99% protein identity. So it really depends. I mean, there's a bunch of different kinds of analysis. We've used, mutation rates. We've used, what can I say, selection, and tests associated with selection to see how they're retained on genomes. But as far as Vibrant goes, I don't think we rely on metabolic genes to a large degree to identify viruses. In fact, I would argue the opposite. We actually rely on structural genes, or what we call viral hallmark genes, more than metabolic genes.
I guess maybe the question was more for the AMGs, to your point that you just said about the evolutionary pattern. I guess it's not clear to me whether you think of AMGs as acquired from the host at one point, and then they remain on the phage and diverse, and therefore you can track this sort of linear accumulation of nucleotides, versus if they're constantly switching and pinging back and forth to their hosts.
So there is an argument that I have seen in literature that there is pinging back and forth between viruses and their hosts, but often I think it's a one-time acquisition, and then if there's fitness benefits, the virus retains it, and then there's vertical transmission. Yeah, I don't think there's a lot. Personally, I think there's not a lot of pinging back and forth there in terms of how this happens.
Great, thanks. Let's, if you wouldn't mind, meet up with people at the break, and then we'll follow up. But one of the things I'll just say that strikes me that I hadn't thought much about, but the viral mediation of these new metabolic traits basically alters transiently the range of the host.
Yep.
And one of the things that implies then is that the virus is revealing one of the main constraints of the range of the host. And so that provides some insights into the microbes' relationship with its ecology and the things that limit it, and the viral effects that relieve that constraint. So I think that's absolutely fascinating at a broader level. Thank you so much.
Thank you.